Okay, let's get started with the lecture. So last time we talked about differential expression, multiple hypothesis testing, multiple hypothesis testing correction, and then gene ontology analysis. Today, there was one section that we didn't quite finish, which is gene set enrichment analysis. So this was first invented in the microarray days, but later on people also use it for RNA sequencing. So there are some experiments when you compare the two conditions, there might be no or very few genes that are significantly differentially expressed if you correct for multiple hypotheses. But it could be that a group of genes that are related to certain functions are only slightly differentially expressed, but coherently. So for example, supposedly we have some sample in one condition and another condition. If you look at all the differential expression, every dot here is one gene, the X and Y axis is the, say, expression level of the two conditions. You might see there are not many genes differentially expressed. There might be two dots here, but they might even be the wrong answer. However, if you look at the genes that are annotated to have the same gene ontology, biological function, or they're annotated to be in the same molecular complex, or they have the same pathway, you might see in this case, these genes that are annotated as sharing one particular gene ontology category are colored red. Did you notice something special about these genes? They are only slightly more expressed in the x-axis and less expressed in the y-axis. If you look at one gene at a time, they are definitely not significant. But if you look at all of those genes together, you might notice a little bit that they are a little bit shifted to the lower right half. And so that's how gene set enrichment analysis is initially developed. And to do something like this, in 2003, the original paper by Vamsi Mutha, who is now a faculty at Harvard MGH, he used this Kolmogorov-Smirnov test. This is a non-parametric test. And so after a gene expression, differential expression analysis, instead of selecting a particular cutoff to say, I want to only have X number of genes at differentially expressed at certain fold change cutoff or at certain FDR cutoff. Instead, you can rank all of the genes, all 20,000 genes from the beginning to the end based on their level of fold change. And so in this case, if you look at all the genes in here, I think this dotted line is the genome-wide distribution, which is a cumulative fraction function, which is what fraction of genes have a fold change below this level. So you might see initially there are some genes that have fold change. By the way, this is already a log fold change. Sorry, this is a fold change. So you might ask what fraction of genes are below 0.1 fold change, which means the treatment condition, treatment and control are tenfold different, right? And you might have very few, but then you ask how many genes are differentially expressed with one fold change, which means there is no differential expression. How many genes are differentially expressed below a 10-fold change? So you can see pretty much all the genes have a fold change below 10. This may not really be the best example because in reality, what percentage of genes do you expect to have fold change below 1? Fold change of 1 means there's no differential expression, right? And so you probably would expect half of those genes. So this may not really be the best example. In the real case, the genome-wide distribution should, at fold change of 1, pretty close to 50%. And in this case, neither conditions are like this. But imagine you use all the genes in the genome to generate that background distribution, and then you ask, If I'm only interested in the genes that are annotated to have a specific gene ontology code and take those genes, for example, I want to look at all the genes related to mitochondria, or I want to take all of those genes that are related to chromatin remodeling, right? So all of those genes annotated with a particular function or complex, right? Then you look at those genes' differential expression and ask what percentage of those genes have a fold change below 0.1, 1, and at every percentage. And then in the process, we ask what is the separation between the genome-wide distribution and the distribution from your gene set. And this D is measured as the maximum difference between the gene of interest, the gene set of interest and your genome-wide distribution. And there is a non-parametric test called a KS test that can give you the p-value of significance. And so this was really quite interesting when people found that after maybe their experiment don't perturb the cell as much, they don't see many differentially expressed genes, but then they can run the gene set enrichment. You just give all the genes a rank order, and then you enter this whole gene set in that order into the this gene set enrichment analysis tool, and then the tool will automatically look at every gene set that shares the same gene ontology code. And then check each gene ontology or Go code whether genes with that Go annotation are significantly different from your genome-wide differential expression level. And so question is, do you need to do multiple hypothesis testing in this case? Yes?
If you run this for multiple specific functions,
yeah,
you're just testing the distribution of a single set of specific functions.
Yes. So the answer is, if you are only interested in a specific gene ontology annotation, then you don't need to do multiple hypothesis testing. Most of the time when people run this analysis, they just provide the differential expression gene order, and they don't a priori know what gene set might be interesting. And so this GSEA tool will automatically test all the gene ontology code. Then you have to do multiple hypothesis testing because it does test 180,000 different Go codes, okay? Yeah, so the user input is all the genes ranked by their differential expression. There's no cutoff. You don't need to set the cutoff. And then the algorithm will automatically check for genes with specific gene ontology annotation that are involved in the coordinate up or down regulation, right? So you evaluate the significance as a whole. The key is to define the gene set before looking at the data. You can't just say, what if I take all the genes, the top 100 genes that are upregulated in my ranked order list and look at their significance? Of course, those are going to be super significant, but you cannot look at the data first before you do this. Right? You have to define the gene set ahead of time and also correct for multiple hypothesis testing. Later on, this tool has been gradually improved and is developed by the Broad Institute between Harvard and MIT. And now there's a tool they evaluate. So you can see the idea is like this. If you look at all the genes that are either upregulated or downregulated genome-wide, this is kind of the genome-wide order. And red is the upregulated genes and the blue is the downregulated genes. And every time you can see this is the genome-wide distribution and the level of differential expression, a pretty smooth curve. And the black bar here indicates the genes that are annotated to contain a specific gene ontology code or whatever gene set that this provides. And so you can see here, there are genes that are more skewed towards the left. Therefore, in the gene ontology analysis, you see that there is something called a leading edge subset, which means that the genes that are annotated to have this particular gene ontology, they are more enriched in the upregulated genes in here. So you can see, if you look at the black bar, they are skewed toward the left. And that means that genes annotated as these are overall more upregulated. Whereas this is another situation, you can see this is the genome-wide distribution. And then in the leading edge analysis, you see that genes that are annotated with this particular Go code, they are more skewed toward the right side, which means those genes in general are more downregulated in here, right? So that will give you a kind of a downregulation enrichment. And so this can be used to evaluate enrichment for both upregulation and downregulation. And since then, the Broad Institute has really expanded this into something they call the molecular signature DB. The paper was published in 2005, but since then they have continued to collect the data and put them in the MSigDB. So the idea is, in addition to genes that are annotated to belong to specific gene ontology as one group, you can also look at the previously published studies and look at those genes that are upregulated. So supposedly you take all of the publicly available expression data, including microarray, including RNA-seq, and each of those experiments, when people are perturbing the cell or comparing the differentiation status or drug treatment, disease, or normal status, and then they look at a set of differential expression, you could use those genes to define a gene set. So instead of it, so in this case, each of the bar can either be genes that are annotated as belonging to the same gene ontology code, or they could be genes from a previously published experiment, are all differentially expressed or upregulated in that experiment, or maybe downregulated in a different experiment. This way, when you are doing an experiment yourself, say you perturb the cell, and then you see some genes differentially expressed, you input the whole gene order into this experiment. And then maybe you go run through the GSEA, it will tell you not only maybe there are some biological functions or pathways that are enriched in your differentially expressed genes, it might also tell you that previously some people have done a similar experiment for differential expression, and those genes that are, for example, upregulated in breast cancer are in your differential gene list that are enriched, right? So in this case, you can use a GSEA to help find other published studies that might be related to yours. For example, if you're treating a cancer cell and you see some genes downregulated, and this group of genes happen to be also those genes that are causing a disease, then maybe that's an important drug you can use to treat the disease, right? That gives you a lot of hints whether your experiments, whatever drug treatment or disease, are relevant to some other published studies. Okay, so that's a very, very useful tool. So the summary here is gene ontology. We mentioned that it's a tree, it's an acyclic tree, and every gene is annotated to have three codes: molecular function, biological process, and cellular components, right? And there are many tools that are useful to help you annotate a group of genes that are enriched in any of these three things. And the one tool that we really recommend is this David tool. You will be using those in your homework, and you can see that it's pretty easy to use and give you some quite meaningful result. And then gene set enrichment is you don't really decide what group of genes to use. You just give it a whole gene order. And then you can use it to detect a general trend without setting the limit on where to cut off of differential genes. And it uses this Kamenov-Smirnov non-parametric test to see whether genes predefined are enriched in your upregulated genes or enriched in your downregulated genes. And here you do also need to do multiple hypothesis testing because you're testing many, many gene sets for either belonging to the same gene ontology group or belonging to another experiment which have shown to be differentially expressed. And since then, this molecular signature DB is part of the gene set enrichment to help define the gene sets, but also bring relevant studies to a user if you have your own differential expression. Yes, question?
I'm really sorry. Can you explain the KS test again, especially the plot?
Here?
Yeah.
Yeah, so basically you have two curves. One curve is to ask what percentage of genes have differential expression below a certain fold change, genome-wide of all the genes. So you can imagine very, very few genes will have a fold change of less than 0.1, which means after the whatever treatment or your differential condition, the treatment and control has a fold change. Basically, it means the treatment will decrease the gene X by tenfold. That is the fold change of 0.1. Then there will be a little bit more genes with the fold change below maybe like 0.5, and then there will be more fold change of below 1. And usually this one is maybe not the best example. Usually you would expect majority of those genes are not differentially expressed. So they should give you a fold change around 1. That's why this is not necessarily the best background. So supposedly this is the background. It should be around 50, where the fold change is 1, and about 50 of the genes are not differentially expressed. Right? Their fold change will be close to 1. But then as you keep going, you ask what percentage of genes will have a fold change below 10? Well, almost none of the genes will have a fold change above 10. Therefore, 100% of the genes are below 10. Right? That's your background distribution. Then you ask, for my gene set of interest, which is defined as sharing a particular gene ontology code, you ask how many percent of these genes have a fold change. So the y-axis is the percentage of genes in this group have a fold change below 0.1, below 0.5, below 1, below 10, and so on. Okay, and so this just asks, is the genes in this gene group very different from your genome-wide in terms of their cumulative differential expression level? And if they are very, very different, that's something interesting, right? You want to know this, even though individual gene may not really show you anything interesting. Okay. Is there another question behind? Yeah.
Oh, I was going to ask what the enrichment scores on the plots.
This enrichment score basically help you evaluate how skewed the gene sets are relative to genome-wide, your distribution. You can see this is kind of like the D-value. What's the best separation between your genome-wide and your gene set of interest?
Does it matter what you actually remember when you look at these plots and you want like a certain shape, you want a certain number?
Usually there is a lead enrichment score you can use, but most people use the p-value or the q-value. Yeah, it's the FDR adjusted p-value is the most indicative. Well, it gives you a rough idea how enriched your data is, but it depends on how big your list is and how skewed it is to the left or to the right. Okay, yeah, another question?
If you don't say something like that, will just tell you how far each other.
So KS test is a non-parametric test, right? Theoretically, if you know your distribution, you can use better statistics. For example, if microarray, you assume that it is a normal distribution after some whatever normalization, then you can use, for example, a t-test as well to look at, because you can see you are asking whether the group of genes you're interested in have a differential expression. You know, if you believe this is kind of a normal distribution, you can use a z-test as well. And so I would say probably over the years, gene set enrichment analysis have gradually also improved their analysis. I'm not sure that this algorithm has been optimized to look at negative binomial distribution from RNA-seq. By the way, a lot of the tools that we discussed in this class were initially developed for microarrays, and people were just continuing to use it for RNA-seq. They are probably decent, but may not really be the best. But a lot of people are now dealing with new type of bioinformatic problems now. They probably are not optimized for a negative binomial distribution. Okay, yeah, another question. Oh, GSEA is gene set enrichment analysis. Yeah. Is to look at, the basic idea is to ask whether the genes annotated as having some gene ontology or pathways are skewed to the left or to the right compared to all the genes. GWAS is to ask if you profile a lot of patients that are disease versus normal, to look at whether some genes are associated with the disease at the DNA level, not at the gene expression level. We will not have this course teaching you exactly how GWAS, and well, we will teach you roughly the basic idea of GWAS analysis, but we'll teach you how to interpret the GWAS analysis. That's mostly looking at, based on your genetic makeup, whether this person is likely to have a disease or not. Whereas in this case, we are looking at the cells in terms of expression, whether the normal versus the disease, maybe even from the same individual, are expressed in a different way. Okay, it's a very, very different type of tests. Okay.